Hello and welcome to this video on meiosis. This is an essential cell function and the means by which we pass on genetics into gametes. This is essential to understanding the many ways inheritance can go wrong. Diseases that have a lot of relevance include those with multiple, missing, or otherwise erroneous chromosomes. Compared to many other things in life, we well and truly understand the process of meiosis. It is a system of changes in a cell that undergoes a scheduled and strictly choreographed process. Deviations and errors are swiftly corrected. The cell can be permanently damaged, or the cell is destroyed. As meiosis uses the DNA directly, it is both a source of intended and unintended mutations. These can be found in the 4 daughter cells that carry only half the number of chromosomes of the parent cell, or occur during crossover when reproduction occurs. Meiosis has 3 regular phases and a 4th technical phase. These are G1, or the growth phase, S1, or the synthesis phase, G2, yet another growth phase, which is replaced by other processes in meiosis, but not mitosis. There is also a G0, or senescent phase. To simplify the understanding of meiosis, we'll draw a comparison between it and mitosis. Meiosis is used for reproductive cells, whereas mitosis is used for those that are not going to be passed on. Fundamentally, the ovum and sperm undergo meiosis, whereas neurons, muscle cells, epithelial cells all undergo mitosis. The first phase, or G1, is the phase in which the cell begins to prepare the various things it will need to divide. This includes proteins, enzymes, and more. It also attempts to incorporate a degree of nuclear material that will be relied on later on. Fundamentally, it is stockpiling. Then we have the synthesis phase. The synthesis phase is where the DNA is replicated. The chromosomes are unwound, the DNA is opened up, and a copy is made. This leaves 4 strands of DNA, or effectively 4 chromosomes. When these 2 are together, they're called a chromatid, and they are attached at the centromere. This is the center line of the cell. Remembering at this stage that the cell has what is effectively 4 chromosomes. This means it has not changed the ploidy, which is whether or not it is a diploid or haploid cell. All cells in the body except those used for reproduction are diploid. If we were talking about mitosis, at this stage, the cells would divide and create 2 exact copies. But we're talking about meiosis here. This means we move on to what is called Prophase I. This is the longest phase of meiosis. It takes the chromosome pairs and attempts to recombine these to slightly shake up the genetic material and create slight differences. This is important in ensuring the next generation inherits both those traits that are desirable, but also creates enough mutations that they can adapt to the new environment. This can lead to chromosomal crossover. That means the different chromosomes can have genes or code move between them. Prophase I is also further divided beyond what we've just described, the first stage of which is called the Leptin Stage. This name means thin threads. This is when the 2 sister chromatids become visible as those thin strands. These are visible within the nucleus, which means the material must have condensed enough, but it also cannot have condensed so far as to have become the chromosomes that we're more familiar with. This is a very brief stage and is followed by what is called the zygotogen stage. That means paired strands, and as the name would suggest, the chromosomes line up with each other to create the homologous chromosome pairs. In this first stage, the chromosomes match up with each other, which means you should get two chromosomes of the same format coming together. These line up in the center, which is where they will begin to be divided. The zygotene stage is followed by what is called the paginated stage. This means thick threads. Generally speaking, at this point, you should find that there is a group of 4 chromosomes formed together. These are in the center, and during crossover, they will normally exchange information, leading to that recombination. At this point, a series of enzymes and DNA checks go through and make sure the DNA is intact, or as intact as possible. It is a far less effective system than that used during normal DNA synthesis, and as a result, errors do occur. It's worth noting at this point that the sex chromosomes do not exchange much information. There is only a relatively small region of homology in which they can exchange DNA or become recombined. This means sex chromosomes tend to be very specific and consistent between parents and children. The next stage is called the diploid stage. It builds on what happens during the paginated stage and slightly relaxes it. This allows for some transcription of DNA, but largely it's there to ensure that the chromosomes remain in the correct place before they go into Anaphase I. This stage is also important as it's where the oocytes or ovum are held until they proceed to the next stage in the reproduction cycle. For women, this only occurs once every 28 days. And that means the oocyte will only be ovulated once, but it will be held effectively in storage until then, with this particular point being where it pauses. Once you begin proceeding on to the next stages, this is where the chromosomes begin to condense. It's called diakinesis. This is where meiosis, or the 4 parts of the chromosomes, which is called a tetrad, become readily visible. The sites where they're crossing over come together and effectively become bound up. This overlapping, almost like a knot, means that you can clearly see certain features. One of these is called the chiasmata. The chiasmata is where that DNA crossover and recombination occurs. At this point, you have all of your chromosomes lined up neatly, and the nuclear envelope begins to dissolve. This disintegration gives the mitotic spindles an opportunity to form. These are essential structures to dismantle or pull apart the various chromosomes. Going from here, we have to enter a multi-stage process to get the chromosomes separated and allow for 2 separate cells to be developed. In order for this to occur, 2 centrosomes are formed. These occur at the polar ends of the cell. These are used to create anchor points. From these, centrosomes are duplicated. These occur during the S phase only. Microtubules then form. These microtubules are important as they're the means, or structurally the means, by which everything is separated out. They then enter the nuclear region after the nuclear envelope begins to be disintegrated and attach to the chromosomes. They bind to a point called the kinetochore. The kinetochore is effectively a little pulley system that allows the microtubules to be ratcheted and gradually pull them apart. Because of the way the microtubules are set up, they are relatively immobile. And so, as the ratcheting moves forward, the microtubules are shifted in a linear manner, forcing the chromosomes to only follow in a specific way. These bind to the 4 kinetocores for each tetrad of the chromosomes. The homologous or matching pairs move together along what is called the Metaphase Plate. This means that the centrosomes attached to each kinetocore will align along what is effectively the horizontal axes. This means the bundle of chromosomes will be split in a way that should create reasonably equal distribution for both force and final product of each chromosome. There is a protein complex called cohesin that holds all these chromatids together, and it will keep them together until the cell can identify that all of the chromosomes have been tensioned using this mechanism. Remembering it is carefully paginated, and if one of the chromosomes is not following the routine, then the entire process is halted until it can be finalized. Now that all of the chromosomes have been tensioned, the cell gives the go-ahead for those kinetocore microtubules to begin pulling the chromosomes apart. Remembering that there's one at each pole, which means the chromosome is pulling to the sides. This allows for the chromosome to be separated as a pair on each side. This means you get 2 matching chromosomes on one end and 2 matching chromosomes on the other, and they remain together due to a variety of special factors that exist in the cell. This is the first meiotic division, and if we were talking about a cell involved in mitosis, this would be the end of where DNA has its actions. But we're talking about meiosis. And meiosis is different because each daughter cell in meiosis only has half the number of chromosomes of a normal cell. At the moment, this process should have produced 2 daughter cells that are still diploid. That means they have 2 full sets of chromosomes as a normal cell would. But the gametes do not. They should only have 1 set of chromosomes or be haploid. Cells undergoing meiosis at this stage will enter a period of recovery. This can be known either as interkineseis or interphase II. But understand, there's been no change to the cell. They should be fundamentally operating on the reserves they stockpiled early on. Meiotic cells now enter a process called Meiosis II. This is the second mitotic division. Unlike the previous process, this is a little bit different. It's now a haploid cell, and there has been no DNA replication. The process here fundamentally takes the DNA in the form of a chromatid, lines them up along the equatorial region, and then begins the series of steps we've described. This is necessary to take the cell from having 46 chromosomes down to 23. We go from being a diploid to a haploid. It has a series of steps involved. Fundamentally, it is the same as what occurs with mitoses, but it has that distinction of being half the number of chromosomes. First is Prophase II. This dissolves the nuclear envelope again. This allows for the chromatids to be condensed and the centromeres to move to the polar regions once more. Yet again, we have development of spindle fibers. Centromeres come out of these and attach themselves to the kinetocores. Then spindle fibers come out and they attach themselves to the centromeres. This creates that equatorial plate. The distinction here is that it's rotated by 90 degrees compared to the previous process. This ensures the cell knows when and where it's working. If it's phase 1 or meiosis 1, the cell knows to rotate it a particular way to get proper separation. If it's aiming for meiosis 2, it must rotate it in order to ensure it's separating it out into a haploid rather than diploid. And then we have Anaphase II. This is followed by a number of steps to cleave the chromatids. They are now called sister chromosomes, and they begin moving away from each other. Then we have Telophase II. Like Telophase I, the chromatids decondense and they become longer. This allows the variety of structures that have been holding them where they are to be dissolved. That means that different parts of the daughter cell that aren't needed anymore and will get in the way of normal function can be dismantled. The cell can go back to functioning in a relatively normal manner for what it is, and you now have 4 daughter cells. Each of these daughter cells is haploid, or only carrying 23 chromosomes. It's important to be mindful of how carefully controlled this whole process is. The intention is to get 23 chromosomes. However, there are some that are more prone to errors than others. A good example is chromosome 21. This is the smallest of the human chromosomes, and being so small, it can often get caught up where it shouldn't be. This can create problems. A triple copy of chromosome 21 can lead to Down syndrome. Having 3 copies of chromosome 13 can cause Patel syndrome. 3 copies of chromosome 18 can cause Edwards syndrome. Then you have sex-specific or sex chromosome-specific problems. Having an XXY or multiple X's and a Y produces something known as Kleinfelter syndrome. Then you have Turner syndrome, in which there is only 1 X chromosome. Then you have 3 X's, which can in some cases lead to excess production of female hormones and other issues related to it. Finally, there's Jacob syndrome, in which it's XYY. That extra Y chromosome in males. All of this is generally associated with an increase in age, both of the mother and father. Generally speaking, the older a mother gets, the more likely there is to be a problem. As you can see, the entire system has to follow that strict process. If there are deviations from what is required to separate the chromosomes out effectively, if the crossover does not follow the right process, if the distribution of chromosomes is not correct when the cells separate, the oocyte can inadvertently acquire mutations, extra chromosomes, or insufficient chromosomes. This does lead to problems down the track if the oocyte in question is the one that successfully becomes fertilized. Thank you for watching this video. If you found it interesting or useful, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions below.